Hello, everyone, and welcome back. It is I, the good Jackson, the goodest of Jacksons, if you will. We are happy to have the whole gang back to get. Are you kidding me? Did I just freeze? Are you for realsies? Okay, there we go. Just kidding. Kidding. Who's Melissa? Joining me today is my host and co-host, as always, Peter. Welcome. And I assume you're off around somewhere now.
I'm kind of backstage-ish.
Okay, that's fine. And then we also have Nestling. Welcome back, who has been on vacation. We had to find other people to fill your role while you weren't here. We talked to an anthropologist. We talked to a data scientist. You know, we did some stuff. We talked to Planet Peterson. Yeah.
Also Aaron as well.
Also Aaron, yeah. So we had some fun. We missed you.
Yeah, you've been busy since I left. Yeah.
Yeah.
I have distracted Jackson with beautiful scientists while you were away.
It's true, and that includes Aaron, by the way.
Yes. Beautiful long-haired scientists. All of them.
I guess people are talking about Florida in the chat. Well, Florida, fun fact, the Surgeon General wants to cancel all vaccine mandates in Florida, which will kill lots of people. Because here's the thing, infectious diseases are a thing. And if you don't get vaccinated for them, they spread. That's how that works. And they cause worse symptoms. They cause a worse infection than you would have otherwise had if you had a vaccine. So it's really dumb stuff. Real dumb. It's because we have an anti-vaxxer who runs our health and human services. And then we have like a COVID COVID crazy guy. I don't know what exactly you call him, like Jay Bhattacharya, who runs our NIH now. And then you have a crazy guy who's Surgeon General of Florida. You have a crazy guy who's Surgeon General of Louisiana. Here in my state, Louisiana, you're not the government-run medical groups that go out and do stuff aren't allowed to talk about vaccines anymore when they go out and do big events. So yeah, no, it's crazy times all around.
By the way, Jackson,
in the toilet.
Have you seen that interview with Kennedy the Crazy that someone put on Twitter, where they were fact checking him in real time? They asked him if he said why he said that there was no vaccine that was safe, and then he said, "I never said that." And the woman interviewer said, "Roll the clip," and so they rolled the clip of him saying that. And then there was another thing, same thing. They just showed him
that is
himself.
Very funny though. They were like,
yeah.
There you go.
Your Honor, it was the brainworm, not me.
That's true. I've seen some hilarious like brainworm memes about RFK. Truly diabolically funny memes.
Jackson, while he may be stupid, and he is, next Monday I will be hosting a show between a guy who's been on your channel, Cypher, and a young Earth creationist who thinks that diamonds are plants. So I'm already clearing out part of my backyard for my diamond plantation because I don't think there's a better way to make money.
Harvesting them like potatoes.
Yeah. So that's going to be a fun conversation.
Yeah, that is pretty wacky. That's one of the wackier claims I've heard. What else? There was something else I was going to say. Oh, he was the last thing I've watched that was involving RFK. I mean, that was like an actual interview with him was when he had his congressional hearing over his nomination for HHS, and one of the people who grilled him but ultimately passed him was a Republican from my state, someone who I have the smidgen or had the smidgenest amount of respect for about 5 minutes, and that is a guy named Bill Cassidy, who is one of our senators. And so Bill Cassidy is a gastroenterologist by trade, so he's a doctor. He understands the importance of vaccines and all that jazz. And so when he was on the panel that was grilling RFK about the past crazy things he said about vaccines and thimerosal and putting chemicals in the water and whatnot, Cassidy said, if I nominate you, if I give you my go-ahead vote, which he ultimately did, he said, you have to promise that you're not going to hollow out our vaccine programs here in the U.S. And Kennedy said, sure, whatever you say. I would never do that. That would be ridiculous. Well, that was a lie. That was what we call in the business a fat lie. So he did do that, and he's continuing to do that. And yeah, so I had a little bit of respect for Cassidy for Dr. Cassidy when he was grilling RFK, but then I lost all of it when he confirmed him anyway. So, all right, without further ado, though, Nestlig, we are on talk 3 of the evolution of photosynthesis, and so I'm going to hand over the reins to you now. So take it away.
All right. And I also there is like one part of my presentation that also there is something related to RFK Jr. or something he said recently, but still, we can begin with the presentation. All right. So this is the 3rd chapter on harnessing light. Basically, we will be discussing how the process of photosynthesis works in excruciating detail, for at least for some people. I think it's very fun, but Jackson told me he wasn't really fond of the at least when he was learning it for the first time, right? You were
yes. When I was in high school, actually, sorry, when I was high school, when I was a grad student, I was like, I hate this crap, and then I became the TA for a botany lab, and I had to learn it and internalize it. Now I teach my own students, and so it's fine now. But back then, I didn't like this. I didn't understand. I didn't like this one or cellular respiration. I hated both of those. But now they actually make perfect sense if you think about it. So,
yes. All right. Next slide. So again, this is the quick overview of the chapters we have done so far. So the first chapter was on the sun eaters, basically a look at the tree of life and which organisms are using photosynthesis for themselves. In the second chapter, the previous one that we did was on how the product of photosynthesis, mainly oxygen, has changed even the very geology of our planet. But today we'll be discussing harnessing light, and the next chapters after, this chapter will basically establish the background knowledge that we will be using to discuss the evolution of photosynthesis, which will be done in multiple parts. Like we have one chapter that will be discussing some parts of photosynthesis, and other chapters a different part of that process. All right, next slide. So again, a quick reminder, the basics of photosynthesis involves this very simplistic chemical formula where you have CO2 plus H2O plus energy in the form of sunlight, and the products will be organic carbon and O2 as a waste product. And respiration, the process that we use in our mitochondria, is the according to the formula, this is the exact opposite of that, but in detail, it is still very different in detail, as we shall see later on. Next slide. So some other let's see here. I have to remember my own slides for sometimes. So there is like in the a couple of 100 years ago. Oh, I see something changing, and okay, can you make it big? Can you make it big? Oh, No. Sorry. Like from our point of view, something changed. Okay, it's back again. Sorry about that. But a few hundred years ago, people were still wondering how plants are able to grow. So you have this one guy called I'm butchering this name, Jean Senabier. He performed experiments demonstrating that plants consumed CO2 and produced oxygen. At first, he described this in terms of the old theory called phlogiston theory. It's an old theory that was eventually replaced by the theory proposed by Antoine Lavoisier, the oxygen theory. Did you know that oxygen is a theory, Jackson?
I did.
All right.
So that means it must not be real. I actually talked about
Yeah.
theories with one of my
Why are you even
classes.
breathing? It's just a theory. Why are you
one of my favorite gags from I think I only saw like 1 or 2 episodes of it, but it's a cartoon called Uncle Grandpa that's very silly. But one of the jokes that they made was he's talking to this like dinosaur, and the dinosaur asks him, "Well, how are you going to get to the moon? What about gravity?" And Uncle Grandpa says, "Gravity is just a theory," and then floats away. And I thought that was very funny.
Speedy in the live chat. Phlogiston is real. RFK Jr. probably said it so on Twitter. Yeah. All right. Also, like he got this part right, that plants consumed CO2 and produced O2, but he got one thing wrong. Senabier proposed that the oxygen atoms in CO2 become O2, but this is wrong. We shall see later. And it's still a common misconception that people think that the O2 comes from CO2 because it's the notation on the formula is very similar right there. But we will see where exactly oxygen comes from later on. Next slide. So later you have another scientist, Cornelis Bernardus van Niel, a Dutch-American microbiologist, coincidentally. It all comes together. He observed the purple sulfur bacteria, which are bacteria
He's
that...
the final boss of the creation-evolution debate. Sorry.
Oh, really?
Well, because he's Dutch and American, so he's the final boss of the creation-evolution debate. Just kidding. Kidding.
Final boss music in the background suddenly begins. Oh, yeah. He observed the purple sulfur bacteria, which is a type of bacteria that do photosynthesis, but they do a type of photosynthesis that does not produce oxygen. Instead of water, they use hydrogen sulfide as one of the starting materials. You can see in the formula on the bottom left right there. So instead of water, you have H2S. And at the end product of this anoxic version of photosynthesis, you end up with sulfur, S2, instead of O2. So based on this observation, he proposed that very similar, when you superimpose the two formulas from the top and bottom, you will see that, perhaps the oxygen atoms in O2 come from the oxygen atom in water. And he was eventually proven right. Next slide. So then you have another scientist in the early 20th century, Samuel Rubin. He also co-discovered the process that produces the carbon isotope 14 with Martin Kamen as well. And what he did was he grew algae with H2O or CO2 enriched in an isotope, an oxygen isotope 18. And based on the tracking where this isotope ended up being, they conclusively prove that the O atom in dioxygen comes from the O atom in H2O during photosynthesis. And the same goes for the oxygen atom that ends up in the organic carbon comes from the oxygen atoms in carbon dioxide. So pretty straightforward, yeah. Also, one tragic thing is that Samuel Rubin died very, very young, at the age of 29. During the early phase of the World War II, he was interested in researching poisonous gases I don't think he was studying them for like as a weapon. He was mainly using to find ways to defend or protect the soldiers from potential exposures. But eventually, sadly, was one accident in the lab, and he was exposed to a deadly dose of phosgene gas, and he passed away from that exposure. Next slide. So that's the very basics of photosynthesis, very easy and very fun to learn. But then when you enter graduate school, then the things get more and more complicated on. Yeah, and today we will walk through the very details of this process, step by step. Next slide. So in our case, we will there are different. As I mentioned before, there are like purple sulfur bacteria. There are also cyanobacteria as well. But we will be using land plants basically as our model to discuss photosynthesis. So we will zooming in on a plant cell. Next slide. And we will be zooming in on the organelle, the chloroplast, where photosynthesis occurs. Next slide. And then there's the chloroplast. There are several compartments and parts, but for now, the main parts that we are looking at are the three compartments: the thylakoid, which is an internal membrane-bound structure, the thylakoid, within the chloroplast. And outside, inside the thylakoid is a space called the lumen, and outside the thylakoid is a space called the stroma. So these are the three main compartments to remember from now on. Next slide. So photosynthesis can be divided up into two main processes. The first process, or the first part of the whole process, is often referred to as the light reactions. And this occurs in the thylakoids, in the internal membranes compartment. And in this case, water, or H2O, plus light energy is used to convert ADP into ATP. And it also is used to convert NADP into NADPH. And the waste product will be oxygen. And that's the summary of the light reactions. But later on, we will be looking at this in more detail. Next slide. And the second phase, or the second half of the process, is called the Calvin cycle. It's sometimes referred to as the dark reactions, although it's a misnomer because the Calvin cycle does not operate without the exposure to light. So even the Calvin cycle is turned off during nighttime, for example. And in the case of the Calvin cycle, it uses the ATP and the NADPH produced from the light reactions to convert CO2 into organic carbon or sugar. And the waste product will be ADP and NADP+, which are then used as the starting material for the light reactions again. All right, next slide. And again, if there are any questions in the live chat, please feel free to interrupt me when I'm speaking.
And the answer to the question on all your minds is, will this be on the test? The answer is yes.
Yes. So, but now when we look even further, at this point it gets very, very complicated. You can see even within the light reactions, there are several different protein complexes responsible for the whole process. And even in the Calvin cycle, there are also several steps as well. But to fully unpack this, we first need to learn a bit of background about the redox chemistry. Next slide. Oh, next slide, Peter. Oh, there we go. Thank you. Am I still audible? Is my mic still clear for everyone?
Yes.
All right. Sometimes my mic acts up a bit, like it did in the last chapter. So
I'll just distract the guests with my juggling if if it happens, if we robot.
All right. Well, thank you. So one thing to remember about the periodic table is that different elements in the periodic table have different affinities towards electrons. Some elements have a strong affinity, and others have a low affinity. And you can see like one very good image from Wikipedia where you can see that the ones that strongly pull electrons to themselves are on the upper right corner of the periodic table, and the ones that have a very weak pull are on the bottom left. There's a very curious correlation right there, and this is a very significant effect on the chemistry of everything, basically. So chemical bonds are either non-polar when when two atoms bond to each other, and if they have the same strength or the same pull strength on the electrons they share, then the electrons are equally shared among them, so they will be non-polar, the bond. And if the strength difference is unequal, then the electrons will be unequally shared, so then you have a polar bond where one atom pulls the electrons to one side, which becomes negatively charged, and the other side will become positively charged. And a very good example is a water molecule, right? So the oxygen in a water molecule is negatively charged, and the H or the hydrogen atoms are positively charged because the oxygen has a very strong pull on the electrons relative to hydrogen. And if the strength difference is even stronger, even more different, then you end up in an ionic bond where all the electrons end up on one atom, and the other atom just loses the electrons. And so redox reactions involve at least the ones that are spontaneous redox reactions involve compounds that with low affinity towards electrons give up their electrons and transfer these electrons onto compounds with high affinity towards electrons. Next slide. So redox refers to reduction and oxidation. Reduction is gaining electrons, and oxidation is losing electrons. And sometimes this gets very confusing because in the formula on the left you can see that the compound A is called the reducer because the reducer donates electrons, and at the end the reducer becomes oxidized meaning it loses electrons. And B is the oxidizer. The oxidizer accepts electrons, and at the end of the process it becomes reduced. It gains electrons. When you learn this for the first time, it gets very confusing to remember what oxidizer and what reducer refers to such. But in any case, the reason why gaining electrons is called reduction, which may be strange because you are gaining something, you're not reducing something. But the reason why is because in historical times, when metallic iron was obtained from iron ore, the iron ore contains oxidized iron in the form of Fe2O3 or Fe3O4. And when this iron becomes reduced, the oxygen atoms are lost, and that's why it's called reduction. The oxygen atoms were lost, and the total mass was reduced at the end of the process of obtaining the metallic iron from the ore. And one common example of a redox reaction is basically burning. When the car burns gasoline, the gasoline is the reducer, it donates electrons, and the oxygen in the atmosphere is the electron acceptor, the oxidizer. In the case of rockets that go out into space, they must carry both the reducer and the oxidizer with them because there is no oxygen in space, of course. Next slide. And everybody still following me on this?
Yes.
All right. And also just in the live chat.
And you have to check with Matt Powell, Neslik, because according to him, there's air in space.
It's just different.
Oh no! Yeah, thank you for reminding me about that. Yeah, it was on your channel, right?
It was on Godless Cranium's channel when I had that conversation because he and Shannon Q, Godless Cranium and Shannon Q, were the ones hosting, and they had me talk with Matt Powell. So that was a fun evening.
Just in the case if there's any audience member not knowing, like Matt Powell does not know, or maybe he does now, I'm not sure. He did not know that the sun does not burn, it undergoes nuclear fusion, and he thinks that it burns something using oxygen in space. Like, okay, and in any case.
Yeah, we can't start listing the things that Matt Powell doesn't know, Nestle, because 2 hours
will be here all night.
Gonna be too short.
All right, so one important aspect of redox chemistry is the redox potential, or the redox potential is measured in volts. It's basically the tendency of compounds in half reactions to gain or lose electrons. The greater the potential, the more stable the electrons are within the compound. So in the case of like the most simple, like one of the very simplest redox reactions is the combustion of hydrogen gas with oxygen. The hydrogen, the H2, has a redox potential of 0 volts. It is the standard. Hence why it is put at 0 volts. But water, or H2O, has an electric potential of positive 0.82 volts. And that means that given the chance, the electrons from hydrogen gas will move towards oxygen, and then the oxygen becomes water combined with the hydrogen as well. And this is the reason why hydrogen combustion is spontaneous and exergonic. It is a downhill reaction when you plot the redox potential on the y-axis on the left, right there. Reducing values are higher, and oxidizing reducing values may be positive, maybe negative, but it is often visual. Like, to help visualizing this process as a downhill process is by inverting the y-axis upside down. So positive values are on the bottom on the graph, and negative values are more higher on the graph, just to visualize electrons going downhill during this exergonic process. Next slide. So the reverse process is still possible, but in this case, it requires an external energy input. So you can perform electrolysis, where you can split water back into oxygen and hydrogen gas. So this is one of my favorite visualizations of what redox chemistry entails, and why one direction is exergonic and why the other direction is endergonic. All right, next slide. So one example of a redox process in biology is what occurs in our mitochondria. So we'll be zooming into the inner membrane region right there. Next slide. So we will zoom in on that. This is this is a very complicated stuff, but we will be just summarizing the broad view of this. So when you eat food, it is glycolysis, and the Krebs cycle uses the food as a substrate, and the products are electron donors. Mainly in this case, the electron donors are NADH on the left end right there. And in the Krebs cycle, you have succinate also donating an electron, becoming fumarate in the Krebs cycle. And these electrons are passed through an electron transport chains, which involves several protein complexes, as well as different compounds with like ubiquinone within the membrane itself, and eventually it ends up in oxygen, the final electron acceptor, which we obtain from air. So this is the very reason why you are eating food and why you are breathing in air at all times, especially the latter part, yeah. And when these electrons are passed through the electron transport chain, this energy is used to pump protons across the membrane. So the protons from the matrix side will be pumped towards the intermembrane side, so from top to bottom, and resulting from this, the intermembrane space will have a higher amount of protons or H plus ions, and the pH will be low as a result, and in the matrix the pH will be high, and this difference in pH gradient powers the very, very famous enzyme called the ATP synthase, which will make ATP using this proton gradient. Next slide. But the energy from this, the energy that it uses is the redox process that I mentioned before with the hydrogen combustion. The electrons go downhill, and that's why the hydrogen combustion is a exergonic process. It releases energy, and it's more or less the same case here. The electrons in the electron transport chain are cascading down. Sometimes they go up sometimes the reaction is uphill, but the total sum of this process is downhill, and that is where the energy comes from that powers our mitochondria, basically. All right, next slide. So now moving on to the light reactions in photosynthesis, it is a very similar, still in detail very different, but still somewhat similar to the electron transport chain in the mitochondria. It also involves a series of electron transfers, and in this case the electron donor is H2O, and the final electron acceptor in this process is NADPH. And we'll be going through the different process itself. Also, one thing to also note is that one protein complex is the cytochrome B6F. It is similar to the complex 3 in our mitochondria. It's a homologous protein. All right, next slide. So when we view this from the perspective of the redox potentials, again, a few slides back, with regarding the mitochondria, the whole cascade of electron transfers was downhill, but in this case it goes uphill in 2 steps and downhill in some other steps. And for this reason, it is often referred to as the Z scheme. Although you have to turn your head 90 degrees.
Not in America, it is. Not in America, it's not called the Z scheme.
It's the Z scheme, yeah.
Uh-huh.
And you have to turn your head 90 degrees to understand why it's called the Z scheme, because it somewhat seems to be a Z almost.
Calling it maths out here with an S.
Yeah. All right. And we'll be going over the individual steps in this process more in detail.
Which I've or an N, you could call it an N. But that is actually how I helped some of the students I had in the botany lab understand what is going on in the in the photosystems. Is pointing out like, hey, this, if you turn it sideways, it's a Z, and then using the arrows and stuff, like it really helps them, because when they're first kind of looking at it, they're just like, what the heck is this?
Oh, Peter, can you go back a few slides? I forgot to mention something that I need to mention something. Can you go back?
Yeah, but how many is a few?
I think 3 slides. Yes, I got it right. All right. So can you see succinate right there, in the middle of it, succinate?
Yes.
Yeah. It donates electrons towards something called FAD. It's I forgot the full name of this. It's a form of vitamin B2. And it is also something that has occurred recently with RFK Jr. I've seen a video of him reading a list of chemicals contained within a food item, and at one point he says riboflavin. I can't even pronounce this stuff. And it turns out riboflavin is a form of vitamin B2, and he doesn't know it, which is very, very painful to see how a
Oh,
public health official does
yeah.
not know what vitamin B2 is.
Or how like carrageenan is derived from seaweed and stuff like that.
Mhm.
John Oliver made a very funny joke in a bit he did on them because they're reading this because RFK, the Secretary of Education, and like a couple other people are reading the names of chemicals off of different foods, and the Secretary of Education is struggling to pronounce one of the names, and John Oliver says, "Yeah, I would like to have a Secretary of Education who can read."
Yeah.
Yeah, that would be nice.
Also, FAD is the full name of FAD is flavin adenine dinucleotide, but it's a very similar compound to like riboflavin is a precursor. If I'm not mistaken, it's a precursor to FAD. Yeah, and this is the reason why we need it, because it's part of the electron transport chain. If you don't get riboflavin or a different form of vitamin B2, then yeah, you're like one symptom is fatigue because you cannot generate the energy from this. All right, so can you go back 3 slides? Yeah, thank you, Peter. All right, so now we continue on with the photosynthesis scheme. All right, next slide. One other thing to mention about this is like sometimes the plant can turn off one of the complexes. In this case, photosystem 2 is turned off, and you end up with a cyclic electron transport where the electrons are going back again and again in a cycle in this transport chain. And in this case, the electrons don't end up in NADPH, so you won't this cyclic electron transport won't produce NADPH, but it will still produce it will still pump protons for ATP synthase to produce ATP. So it's basically a way to if a plant does not need NADPH, but it needs more ATP, then it will go into this mode to produce more ATP and less NADPH. All right, next slide. So, okay, so now we will go to the first step in the process. So we will zoom in on photosystem 2. Photosystem 2 is, like you can see, it is the first protein complex in the chain, but it just happens that the photosystem 1 on the right hand side was discovered before photosystem 2 was discovered, hence why it's called 2. All right, next slide.
And confusing generations of botany students.
Right. So this is photosystem 2, at least the what's called the C2S2N2 type supercomplex of photosystem 2 LHC2. Yeah, it's a very complex found in most land plants. Like I got this model from Pisum sativum. It's the pea plant, and if you are interested, you can find this structure yourself online on the RCSB website. Just put in the PDB ID number, and the number is 5XNM. Just putting that in, and you can find this structure. You can also find various other structures as well if you're interested. It's a super complex for a
Peter
reason.
is dying out here, apparently.
Yeah, I heard that. Yeah. No worries.
No, Peter, you can't die on stream. That would not be good for us.
I was trying out, but Nestle said earlier that we swallow food and we breathe in air. I tried to do it the other way around, and I think he's right.
It happens.
Yeah. All right, next slide. So viewed from above, this is what the super complex looks like. It's basically a radially symmetric dimer. So you have like a repeat of 2 super complexes basically. So on the I drew a white line to denote the symmetry. And the symmetry is in the right in the center right there. And it is radially symmetric. The proteins on top of the white line is repeated, but in the clockwise or 90 degree or 180 degrees turned upside down basically. Yeah. So this super complex is the dominant super complex found in land plants, as I mentioned before. And this complex is found, is the structure that appears when the plant is adapted to limited light. So the structure changes in response to light intensity, which is very interesting. All right, next slide. So there's several different components. So the first few components are minor antenna proteins, which are each is monomeric, so only one polypeptide consists of each of these subunits. So you have the CP24, CP26, and CP29. All minor antenna proteins are nuclear encoded. So again, remember that in plants, you have the mitochondria, you have also mitochondria, but we will ignore the mitochondria from now on. You have the chloroplast and also the nucleus. The nucleus contains a nuclear genome, but the chloroplast also contains its own genome. So you can have proteins encoded in a nuclear gene or also in a chloroplast gene as well. But in this case, these proteins are nuclear encoded. The genes are found in the nucleus or in the nuclear genome. Next slide. So next we have the major light harvesting complexes. These are trimeric. So each of these complexes are composed of 3 subunits of polypeptides. You have different versions of this. You have the strongly bounded S type, the moderately bounded M type. This moderately bounded M type, it detaches from the complex when light intensity gets too high. And if light is very limited, a third trimer called the LC2L type may join as well. But I'm not showing this third complex in this image. And these light harvesting complexes are also nuclear encoded. And they are also one interesting thing to mention is these LHC2 complexes are the most abundant membrane proteins on the planet by far. Like about half of the proteins found within the membranes of chloroplast are these proteins by mass. All right, next slide. So next up we have the photosystem 2 core, or the PES2 core for short. They consist of about 20 protein subunits, most of which are chloroplast encoded. And also unlike the previously mentioned antennas, the LHC, etc., the PES2 core is very conserved across all photosynthetic organisms. So the light harvesting complexes are mainly found in the land plants and also the green algae as well. But when you go into like the red algae, or especially in the cyanobacteria, or even the purple sulfur bacteria, you don't have these light harvesting complexes. You have different types of light harvesting complexes. But the PES2 core, they are there. They are very conserved across all different sun eaters. All right, and these cores contain parts of the electron transport chain that I showed you a few slides ago. Next slide. Also, one thing to mention about these complexes is that they are chock full of these pigments, like chlorophyll A, obviously, also chlorophyll B, chlorophyll B, beta-carotene, and also a few xanthophiles. These, like chlorophyll A, is the main pigment, but you also have these accessory pigments of chlorophyll B, beta-carotene, and the xanthophiles, which will increase the bandwidth, which allows plants to capture more light energy. And also interesting is that the light does not have to strike the PES2 core directly. It can strike a light harvesting complex, as it's shown here, and then the energy is transferred from one pigment to another pigment via resonance energy transfer, and eventually it ends up in the PES2 core, where it does its work. Unlimited power, yeah.
That is interesting. That's really interesting.
Yeah. All right, next slide. So now let's zoom in onto the PES2 core. Next slide. So this is a PS2 core. Like the PS2 core itself is also a dimer. It consists of a copy of 2 complexes, but right now we are looking at only 1 copy of the PS2 core. And in this case, I'm using a model from a different from a bacterium, I believe, yeah, because it's the best model available, at least the one that I found. But in any case, it's still very similar to let's see here. Let's see. Thermosticus. What's that? Is it a bacterium? I think it's a bacterium. Or maybe it's a cyanobacterium, I think. Sorry about it. I'm just trying to fact check myself live in the presentation. Is it a bacteria? Is it a cyanobacterium?
Thermosticus. Do you want me to look it up?
If you can help.
Survey says, that doesn't make it easy at all. Weird.
Yeah, it's a cyanobacterium. Yes.
Oh, yeah, there it is. Okay, I do see it. Yeah, thermophilic cyanobacterium. Wow, that was.
Yeah.
There's no like Wikipedia page for it, which is normally what pops up.
Exactly. Yeah. So one thing to mention is that there are several different components to this complex itself. So you have a few intrinsic subunits. These intrinsic subunits are membrane bound. They're the gray ones, I call it, and most are chloroplast encoded. You also have a few extrinsic subunits. So these are outside of the membrane, and these are yellow, and these also most are nucleic encoded. Although in this case, the extrinsic subunits are not very conserved. Like in the ones in cyanobacteria are different than the ones found in land plants, for example, or at least a few of them are different in land plants. But regarding the last few of them, these are the most conserved subunits. Next slide. So these are these 4 ones are the most conserved subunits. So you have 2 inner antennae proteins, the CP43 and the CP47, and also the reaction center proteins, the D1 and the D2. And they are all 4 of them are chloroplast encoded proteins. And again, highly conserved. They are found in cyanobacteria as well, in a very conserved form. All right, next slide. And within them are the several compounds which we call redox centers, because these are involved in the electron transfers, the redox reactions that I mentioned before in the electron transport chain. And most of these are bounded in the D1 and the D2 complex, in the D2 subunits. Next slide. So the first complex we are dealing with is called the P680 cofactor. It's composed of like sometimes the P680 complex or center is described as a dimer of chlorophyll A, but in our case we will view this as a tetramer of chlorophyll A. So there are 4 chlorophyll A's that compose this center. So you have 2 of them on the left, which are ligands of D1, and 2 on the right, which are ligands of D2 subunit. And also one thing to note is that you have like a few electrons, a pair of electrons in the ground state, but when light energy excites this complex, next slide, then one of the electrons go into a higher energy state, and then this excited state we denote this as an asterisk with an asterisk, the P680 asterisk complex. And when this complex becomes excited, the redox potential changes. It becomes very negative, or on the graph, on the bottom left corner right there, on the graph, it goes higher on the graph. It becomes more reducing, meaning it's more capable of donating an electron to the next redox center. Next slide. So this P680 complex donates an electron to what's called a thiophytin compound. Thiophytin is basically identical to a chlorophyll A molecule, except that thiophytin does not have a magnesium. Oh, is it magnesium? Yes, I think right. Chlorophyll A contains magnesium, right? Just making sure that I'm getting correct.
Should I cue the juggling?
Yes, chlorophyll A contains magnesium. I got it correct. Yeah. But thank you. The thiophytin compound is just a chlorophyll A molecule without the magnesium within the ring, basically. All right. So and then when it donates this electron, the P680 becomes a positively charged radical. It has only one. It has a one unpaired electron, which is a very problem. Next slide. Next slide. Hold on. We will. Oh, sorry. I got my slides in my head wrong. So we will move on to the next slide. Sorry. Yeah. Next slide. We will come back to this radical. We will come back to the P6. No. Go further. Sorry. Yeah, go further. Yeah, thank you. We will come back to the P680. Like sometimes I am anticipating one slide, but it goes to a different slide because I cannot memorize all my slides in my head. So sorry about that. All right. So now,
well, how embarrassing! Can't relate. No, just kidding.
Yeah. The thiophytin donates the electron to the next redox center, the plastoquinone in the QA position. Although this is an error of mine. I notated that the QA is in the D1 subunit. It's actually the D2 subunit. So this should be changed. So instead of saying plastoquinone in the D1 side, it should be saying the D2 side. All right. Next slide. And then this quinone donates the electron onto another quinone, but in this case, the plastoquinone on the D1 side. And again, this is a similar error. It says D2 here, but it should say D1 on the D1 side. This. All right. And this particular transfer of electron is also mediated by the presence of a bicarbonate and also an iron ion as well between these two plastoquinones. All right, next slide. So now we go back to this P680 radical. It is like when after it has donated an electron, the electron left behind is an unpaired electron, and we call this unpaired condition a radical. And this P680 radical is the strongest oxidizer known in biology, with a redox potential of positive 1.25 volts. And this is and if you remember from a few slides back, this has a higher electron potential than water itself. So it is able to pull electrons away from water. This complex.
That's pretty nuts.
Yes. All right, next slide. So but there's a problem. P680 is only able to take one electron at a time. But when you oxidize O2 or H2O into O2 one electron at a time, this will create unstable intermediates, which we call reactive oxygen species. So you can see some of the intermediates right here. So you have like a hydroxyl radical, very dangerous. Hydrogen peroxide, which is less dangerous. Superoxide is also a pretty dangerous radical as well. And eventually you end up with O2 if you manage to pull all 4 electrons. So in order to fully oxidize 2 water molecules into oxygen, you need to pull away 4 electrons, and you need to do that quick, because otherwise the radicals can wander off and cause damage. All right, next slide. But this problem is solved by a very interesting reaction redox center called the oxygen evolving complex, also called the water splitting complex. That has several different names, but the most common names that I've seen is the oxygen evolving complex. It is a complex involving, let's see here, 4 magnesium atoms, 5 oxygen atoms, and also 1 calcium atom. A very complicated mineral structure almost, yeah. And this complex is ligated by the D1 subunit and also the CP43 subunit as well. All right, next slide. And exactly how this reaction center operates is also very complicated. It undergoes several different changes, which we refer to as the within the Koch cycle. You have state S0, state S1, S2, S3, and also S4 as well. The exact structure of, like you see an image right here, which proposes this particular configurations, but the S4 structure is still a bit mysterious. So the scientists are still figuring out exactly how the S4 structure looks like. I won't go into the exact details of how the Koch cycle works. So I could just give a summary on the top right there. The oxygen evolving complex works mainly via the manganese, switching between different oxidation states, the oxidation state 3 and the oxidation state 4. So when magnesium goes from the third oxidation to the fourth oxidation state, the complex donates electrons. And when they go into the opposite direction, when the magnesium 4 becomes magnesium 3, then the electrons are gained from water. And in this way, you prevent the production of reactive oxygen species. The water intermediates are contained within this complex during this cycle. It holds on to the intermediates basically before it releases the oxygen as the waste product at the end of the cycle. Also, one trivia, the Koch cycle, it refers to a scientist whose surname is Koch, and it happens to be a Dutch scientist as well. And if you also, interesting thing is that Koch is an occupational surname. It means chef in Dutch, which seems very fitting because this process makes all of the food that we eat in the world.
It's always the Dutch.
All right, next slide.
That is interesting. So, question is that, and this may be a dumb question, but is that why it's often pronounced cook in America?
I'm not sure. I think it's just the pronunciation of cock is not common in English.
Okay.
So I just think it's the way. I think it's the way that the English-speaking people pronounce cock, because it's a very unnatural vowel in English, I believe, if I'm not mistaken. Yeah. All right. But then summarizing this, 2 water molecules interact with the oxygen evolving complex. The products are is 1 oxygen molecule, also 4 protons, basically hydrogen ions, H+, are released into the lumen of the chloroplast within the thylakoid. The lumen is the space within the thylakoid. And then a full cycle of this Koch cycle donates 4 electrons, 1 electron at a time, towards P680. So and that resolves this problem. P680 is only able to receive 1 electron at a time, but the oxygen evolving complex can donate 1 electron at a time, eventually completing the full cycle of the Koch cycle. All right. And also one other redox center in between is a tyrosine side chain of the D1 subunit that is also involved in the electron transfer. All right, next slide. So going back to the plastoquinone on the again, this also this is the same mistake before. I denote that this plastoquinone is bound by D2, but it should be D1. That's one mistake. It should be D1. So this plastoquinone bound by D1, when it accepts one electron, it becomes a radical, a negatively charged radical. But when it accepts two electrons, or the second one, then two protons, or H plus ions, are from the stroma side. Again, the stroma is the outside space of the thylakoid. So two protons from the stroma side are become bounded to the plastoquinone, transforming it into plastoquinol. And the plastoquinone is notated as PQ, but plastoquinol is notated as PQH2 because the H2 or two H atoms are added onto this redox center when it receives the second electron. Also, when it becomes this PQH2, it is released from the D1 subunit, and it moves within the membrane. It's the quinol is a fat soluble compound. It moves within the membrane of the thylakoid itself, and when it leaves the D1 subunit, another PQ fills in its place, basically. A new PQ will occupy the QB spot. Yeah. All right, next slide. So just a summary of the PS2 process. So yeah, first you have water becoming oxygen and releasing protons within the lumen site. And at the other side, at the end of the process, protons are taken from the stroma site, and then they end up within the PQH2 compound, and a PQ molecule fills in the empty space left behind when a PQH2 leaves the PS2 complex. And as you can see also one thing here, because on one side, in the lumen side, protons are released, and in the stroma side, protons are taken away. This increases the proton gradient, which powers the ATP synthase later on. All right, next slide. So now the PQH2 molecules, they will move within the membrane itself, the thylakoid membrane, and they move on to the next protein complex, the cytochrome B6F complex. Next slide. So this is also a radially symmetric dimer. Viewed from above, you can see it's radially symmetric. So there are two copies of complexes, but we will only look at one to make it simpler. So next slide. So this is only one of the two duplicates, and it is also composed of several polypeptides. So you have four small polypeptide subunits. One of them is nuclear encoded, but we have four main subunits. The cytochrome B6 subunit, which is chloroplast encoded. The cytochrome F subunit, also chloroplast encoded. You have subunit 4, also chloroplast encoded. And you have a subunit called RISC, which is a nuclear encoded polypeptide. All right, next slide. And the redox centers are ligated by the four large subunits I just mentioned. These are involved in the electron transport chain. Next slide. All right, so when a PQH2 molecule goes into this cytochrome B6F complex, it undergoes a process called electron bifurcation. So remember that this PQH2 molecule carries two electrons. And when it donates the electron, the two electrons go into separate paths. One of them goes downhill energetically, and the other one goes uphill energetically. And the reason why the electron that goes uphill can go uphill is because the energetic cost is more, in a crude language, it is paid by the other electron going downhill as well at the same time. It's a mechanism of energy conservation. You release energy, or you release more energy in one half of the process, and then it allows you to increase energy in another half of the process. I need to get some water. Be right back, real quick. You can juggle the animation.
You want me to juggle? Okay. This has been really interesting so far, and yeah, I really came to appreciate photosynthesis, and I think its evolution is really interesting. I would love to read that book that Nestling mentioned by Nick Lane early on, but I have not. I've still only read The Vital Question by Nick Lane, although really I haven't read much of anything recently because I've been so busy writing lectures for my classes. And I don't even have as many classes as my wife does. She has five classes, I think, which is a lot. It's too many, in my opinion. But I just have 2.
All right, I'm back.
Welcome back.
You can continue on explaining something.
You're good. It's all good. I was just filling space.
Yeah. But as I mentioned, that the electron bifurcation is like an uphill and a downhill process at the same time. And because the downhill process happens at the same time, it allows one electron to go uphill energetically, basically. All right. And also the protons that are released from this electron bifurcation, it is released into the lumen side again. The space within the thylakoid. All right, next slide. So the electron that goes uphill via one redox center called cytochrome B6, or it's specifically called the CYTBL redox center, and then it goes on to another cytochrome B6 compound called the CYTBH, and then it ends up in a heme C compound called CYTCI. So you have different hemes basically as the redox center, heme B, and the heme C compound. They are similar to the heme that we find in our hemoglobin. Not identical, but it's a chemically similar compounds. I'm not sure. Like, what is the heme type found in our hemoglobin? I'm also curious.
I don't remember offhand. You'd have to look it up.
Yeah. Hemoglobin type found type of heme type found
heme or her,
oh. All right, let's see here. It's a heme B. All right, so it's the same type. It's the same type that we see here, basically.
That's what I was thinking.
Yeah, heme B is a type found in hemoglobin, but it is not involved. That heme in hemoglobin is not involved in electron transfer. It's involved in, of course, carrying oxygen. Yeah. But in any case, the electron will end up in another PQ molecule as well, which will get one a proton from the stroma side. But when we perform another electron bifurcation process, next slide. Then what we end up with is the top side, the PQ on the top side, can obtain 2 electrons, which will become a PQ2 as well. So what we end up with is a cycle, what we call the Q cycle. It basically allows the extra energy from electron bifurcation to pump more protons away from the stroma into the lumen side. It's a very new. It's basically you get more from your PQ. You get the most from your PQA H2 than you would have otherwise get. All right, next slide. So on the other side, the PQ cycle is involved in the electron transport chain of one side of the electron bifurcation process. But the other side of the electron bifurcation process, the electron goes through this F2S2 redox center with basically 2 iron and 2 sulfur atoms within the RISC subunit. And then it goes on to another heme C compound within the cytochrome F subunit. And eventually it ends up within the electron ends up within another protein called the plastocyan cyanin protein, and this has a copper atom which will accept the electron from the cytochrome B6F supercomplex. All right, next slide. And this plastocyanin is water soluble. It will like after it has accepted the electron from the cytochrome B6F complex, it will just move around within the lumen. And again, we also see that the cytochrome B6F, it has taken away protons from the stroma site and increased the protons within the lumen site, contributing to the proton gradient that powers the ATP synthase. All right, next slide. And then the plastocyanin, it is water soluble. It will just float around within the lumen, and then it will end up binding onto the next supercomplex, the photosystem 1. Sorry, almost said 2. Photosystem 1 complex. All right, next slide. So viewed from above, it is not as big as the supercomplex of photosystem 2, but still pretty impressive. All right, next slide. It has various minor subunits. Most of these minor subunits in gray are nuclear encoded. You also have 4 light harvesting complexes in purple. These are also nuclear encoded. But the PS1 core is mostly chloroplast encoded. So now next we will zoom into the PS1 core. Also the PS2 core that you see here is also a dimer. There are two copies of the same complex binding to each other radially symmetrically. All right, next slide. Oh, also the same process also also applies here. You have chlorophyll A and also chlorophyll B, and also beta carotene, xanthophylls, pigments, which will also increase the ability of light capture. And also the same principle applies here. The light energy doesn't have to strike the core directly. It can first strike a light harvesting complex, and the energy can be transferred from one pigment to another, eventually ending up in the core of the PS1 complex. All right, next slide. So now we zoom into the PS1 core. Next slide. So now we see that. I think I got something wrong. The core is not a dimer. No. In the case of the PS2, it is a dimer, but in this case, it's not a dimer. All right, so never mind. The PS1 core, and we can view it here. It's composed of 4 main subunits, or in this case, yeah, we will include the plastocyanin as a subunit. Like it can detach. It often detaches from the complex, of course, but right now we will just view it as a subunit. So you have the plastocyanin on the bottom, which is nuclear encoded. We have 2 main reaction center proteins, PSA-B and PSA-A, both of which are chloroplast encoded. And on the top of the complex, we see the PSA-C subunit, which is also chloroplast encoded. And these, next slide. And these subunits contain the redox centers found within the PS1 system. Next slide. So the first one is the first redox center found is called the P700 complex, which is a dimer. Two chlorophyll A molecules. Although one of the chlorophyll A molecules, which is ligated by the PSA-A subunit, it is a modified version of chlorophyll A. It's a stereoisomer, specifically a C132 aptamer of chlorophyll. The details you don't have to. It's not on the test. Let's say exactly what that means. All right. And also, this P700 is ligated by both the PSA-B and the PSA-A subunit. So one of the 2 chlorophylls is bounded by PSA-B, and the other one is bounded by the PSA-A subunit. Next slide. And when light energy strikes or indirectly strikes this PS700 complex, it becomes excited. It's the same principle as the P680 complex within the PS2 system. It also becomes excited. All right, next slide. And the electrons can be donated on to the next compounds. And in this case, I think I forgot to mention this aspect of in the PS2 system. In the PS2 system, you have the P680 complex. It donates the electrons onto a pheophytin molecule. But there are two pheophytin molecules in the PS2 system. But the P680 will only donate electrons to one side, not the other one. Only one of the two pheophytin molecules. So it's a different. So it's only one option. But in the case of the PS1 system, it can go both directions. So it can go on the left hand side or on the right hand side. The electrons can be donated. It's symmetrical basically. One electron can go in one side or on the other side, but it will eventually end up in the same place regardless of which side it is donated. So from the P700, electrons are donated to another chlorophyll A molecule within the A-1b or the A-1a. And the next is another chlorophyll in the A0b or the A0a position. And then the next electron of the next redox center is within the A1b position. It's a phyloquinone molecule, a form of vitamin K1. That's another vitamin involved in electron transport chains, and that can donate the electrons onto an Fe4S4 complex, another iron sulfur cluster within the Fx position, and this Fx is bounded by both PSA A and PSA B subunits, and eventually the next redox center is the next redox centers plural are found in the PSA C subunit. So you have then the Fa iron sulfur complex, and then finally in the Fb position, another iron sulfur complex is the final electron or the final redox center within the PS1 system. All right, next slide. And again, after the P700 has donated the electrons upwards in this image, then the plastocyanin can donate its electron onto P700, filling the empty spot of the after the P700 has donated an electron, there is an empty spot within itself, which is filled up by the electron donated from the plastocyanin protein. All right, next slide. So the PS2 eventually it's not we are not done yet. The PS1 protein complex will donate the electron onto another protein called ferredoxin, which has an F2S2 iron sulfur complex. And next slide. And that ferredoxin eventually donates the electron onto another enzyme called the ferredoxin NADP reductase. It has another instance of the FAD, another instance of the FAD cofactor, the vitamin B2 that I mentioned regarding RFK Jr.'s comment or about riboflavin. It's the same compound here as well. When it accepts the electron, or when it accepts 2 electrons from ferredoxin, the FAD becomes FADH2, and that can donate electrons onto the final electron acceptor of this process. Next slide. So the final electron acceptor is the NADPH. So FADH2 will donate electrons onto NADP plus, and that will become NADPH. And that is required to drive the Kelvin cycle, as we will see later on. And also one thing to mention is the like when NADPH plus becomes when NADP becomes NADPH, it will gain the proton from the stroma side. All right, next slide. So this is the summary of the whole process we have discussed so far, from the light dependent the light reactions. First, we have water becoming oxygen. Several things happen. Protons are removed from the stroma side and given into the lumen side, contributing to the proton gradient, powering ATP synthase. And eventually the electrons end up within the cofactor called NADPH, which is needed for the Kelvin cycle. All right, next slide. So again, this is the summary of what we have discussed, but now we view it in the with regard to the redox potential. All right, next slide. All right, and now we move on to the Kelvin cycle. Again, summarizing, the light-dependent reactions produces the NADPH, as discussed before. The proton gradient produced by proton pumping powers the ATP synthase, producing ATP, and the ATP is also required for the Kelvin cycle. The Kelvin cycle is also called the CBB cycle, referring to the three scientists called the Kelvin, Benson, and Bassan. So that's three different scientists that they've all contributed to the discovery of the Kelvin cycle, but it's more often called the Kelvin cycle since it's more simpler than the CBB cycle, more easier to remember. All right, next slide. So the first step of the Kelvin cycle is what's called carbon fixation. So carbon dioxide is assimilated into biochemistry, basically. And then the enzyme responsible is a very interesting enzyme. It's called the Rubisco enzyme. The full name is ribulose bisphosphate carboxylase oxygenase, or Rubisco for short. And it's also the Rubisco enzyme is composed of 8 large subunits and 8 small subunits. And the large subunits are chloroplast encoded and the small subunits are nuclear encoded. Also interesting to mention it is that the Rubisco is by far the most abundant protein on the planet. Like, I think about 43% of the protein mass contained within leaves of any plant is Rubisco. 40% of the protein within a leaf is just Rubisco. Yeah. I will discuss Rubisco in more detail in a future episode when we discuss its evolution. So, but for now we have the substrate of Rubisco is a 5-carbon compound called ribulose bisphosphate, so RUBP. And when Rubisco attaches the CO2 onto this 5-carbon compound, it produces a 6-carbon compound intermediate, but that is an unstable one. It will spontaneously break apart into 2 identical molecules of 3-phosphoglycerate, or 3-PG. All right, next slide. I also, in the diagram on the left, I also notated the number of carbon atoms per molecule, so you can follow it more easily. So again, we first started with the 5-carbon compound, and now we end up with 2 3-carbon compounds. And the next step involves the enzyme phosphoglycerate kinase, also nuclear encoded. It will convert the 3-carbon or the 3-carbon compound and another 3-carbon compound, but now it will add a phosphate onto the compound. And this consumes ATP, becoming ADP. All right. And you can see also the structure of ATP on the left-hand side. ATP has 3 phosphates, and when it becomes ADP, it loses one of the phosphates. All right, next slide. All right, the next step involves the enzyme glyceraldehyde 3-phosphate dehydrogenase. And this will convert, or this will reduce the previous compound. Again, we have the previous compound is the, I won't pronounce the full name, it's just a 3-carbon compound with 2 phosphates. And it will reduce this compound, basically adding a hydrogen atom onto, and also an electron with it onto the compound, and hence why it's called reduction. When you add electrons to a compound, the compound is said to be reduced, and the reduced product is glyceraldehyde 3-phosphate, or GAP. And it's a 3-carbon compound, and during the process the phosphate is lost. So now it only has 1 phosphate group in the molecule. All right, next slide. So this is the end product of, or at least one of the end products of the Calvin cycle. So we have 2 GAP molecules. One of the 2 GAP molecules can leave the cycle and become the building block to build biomass. But we have a problem. We need to reproduce or regenerate the starting material in order for the Calvin cycle to continue. If it doesn't get replenished, the Calvin cycle just stops. So there's right now we have one 3-carbon molecule to be used, but that's not enough to regenerate the 5-carbon molecule that we need at the beginning. So what we need is next slide. The cycle needs to be repeated 3 times in order to have enough material to regenerate the starting material. So now when we repeat the previous processes 3 times, we end up with 6 GAP molecules. One of the GAP molecules can leave, so we still have 5 GAP molecules left over to replenish the starting material. And if you can count the number correctly, 5 times 3 equals 3 times 5. So we can thus use 5 3-carbon compounds to regenerate the 3 5-carbon compounds that we used at the beginning of the Calvin cycle. Following with me so far?
Yes.
Yeah, all right. This is on the test.
Sorry, I had my mic muted, but yes.
All right, next slide. So to regenerate the 5-carbon compounds from the 3-carbon compounds, we need to do several steps. Like first, 2 of the 5 GAP compounds are converted into molecules we call DAP or DHAP. And this conversion is mediated by an enzyme called the triose phosphate isomerase. So now we end up with 3 GAP molecules and 2 DAP molecules. All right, next slide. One of the GAP molecules and one of the DAP molecules are combined via an enzyme called aldolase, which combines the 2 3-carbon compounds into a 6-carbon sugar called fructose 1,6-bisphosphate. So basically fructose with 2 phosphate groups. And also, next slide. And then another enzyme called fructose bisphosphatase. This will remove one of the phosphate groups away from the fructose. So now in the first we have a fructose with 2 phosphates, and now we end up with a fructose with only 1 phosphate attached to it. And this is one of the irreversible reactions. Like many, I should have mentioned this before. I in the right hand side, I put an arrow beneath the enzyme. If the arrow has 2 arrows on both sides, it's a reversible process. The reaction can go backwards, but in this case, the reaction can only go forwards. It's an irreversible reaction. And there are a few of these irreversible reactions within the Calvin cycle, which allows the Calvin cycle to go into only one direction as a whole. Yeah. And also, one other thing to mention about this fructose product, this fructose can also leave the Calvin cycle and be used as a precursor to make starch. But in order for, but again, if we take away this fructose, then the Calvin cycle cannot be replenished. So in order for the Calvin cycle to be replenished, you have to repeat the Calvin cycle 6 times. And that allows 1 fructose to leave the Calvin cycle if you repeat the Calvin cycle 6 times. All right, next slide. So next up is if we use the fructose to regenerate the starting material, then the fructose is used by another enzyme called a transketolase. Basically, the 6-carbon compound, or the fructose, donates 2 carbon atoms to a 3-carbon compound, in this case another gap molecule. So the gap molecule gains 2 carbon atoms, and the fructose loses 2 carbon atoms. So on one end, we end up with a 5-carbon compound called xylulose 5-phosphate, or XU5P. And we also end up with a 4-carbon compound E4P. Next slide. And now we also another aldolase reaction is involved here. The 4-carbon compound previously mentioned is attached onto another DAP molecule by the aldolase enzyme. I believe it's the exact same aldolase enzyme that is involved in a few steps previously as well. It's an identical molecule, or at least a similar enzyme, if I'm not mistaken, if not identical. I think I will discuss this more in detail. Like, I will discuss how the enzymes are related when we discuss the evolution of the Calvin cycle in another episode. Yeah. So right now I'm not remembering exactly what the relationship is. Yeah. All right. So in this case, the aldolase combines the 4-carbon compound with the 3-carbon compound, producing a 7-carbon compound with two phosphates. And that is we call it the S17BP abbreviated. It is a very long name. When you see it on the bottom right there. I won't pronounce that. Next slide. So then another enzyme will remove the phosphate from this 7-carbon compound, and this is something I do remember. Like in a few steps before, we have the on the Calvin cycle we have the FBPase, which removed the phosphate from fructose. That enzyme is related to this enzyme, the SBPase. It performs the same. It performs the same reaction, except in the previous case, a phosphate is removed from fructose, a 6-carbon compound, but here a phosphate is removed from a 7-carbon compound. Very similar reaction, and homologous enzymes. And if I remember correctly, in land plants we have these 2 enzymes, homologous, but in cyanobacteria they are identical, the same enzyme. It's not duplicated in cyanobacteria. It later was duplicated in the land plants, which gave rise to these two different enzymes, the SBPAs and the FBPAs. All right, next slide. Just a little teaser for what we will discuss regarding the evolution of the Calvin cycle. All right, so the next step is another transketolase reaction. The 7-carbon compound previously mentioned will donate 2 carbon atoms to another 3-carbon, the third gap molecule left over from the previous reactions. So the 3-carbon compound gap gains 2 carbon atoms, becoming a 5-carbon compound. And when the 7-carbon compound donates 2, it becomes a 5-carbon compound as well. Slightly different. They are like one is XU5P, and the other one is R5P. It's not identical, but still it's the same number of carbon atoms contained in these two. All right, so next slide. So now we have, at the end of this, we have 2 of 3 5-carbon compounds. 2 of them are XU5P, and 1 is R5P. And these have to be isomerized to produce the correct 5-carbon compound. So R5P and XU5P are converted into RU5P, or ribulose 5-phosphate. And there are 2 enzymes involved in this isomerization. One of them is the phosphopentose isomerase, and another one is the phosphopentose epimerase. Let's see here. All right, yes. And now the final step, next slide. And this ribulose 5-phosphate, another enzyme called the phosphoribulokinase, this will add a phosphate group to this 5-carbon compound. So now previously ribulose 5-phosphate only has 1 phosphate group, but at the end of this process, the 5-carbon compound has 2 phosphates. And again, this will consume an ATP, leaving an ADP as a leftover product. And now we have successfully replenished the starting material so this allows the Calvin cycle to continue on again and again and again. All right, next slide. And that's it. We have fully discussed the process of photosynthesis. So next up, we will. I will be focusing on the evolution of reaction centers that we discussed in this episode. But I'm not sure exactly when we will do that. I have not made even a single slide yet, so it may take like many months before I'm ready to do that.
Hey, that's fine. You take as long as you need, to make it cool and informative and everything. Okay.
And we finished pretty early this time. I'm very surprised that I was able to finish like 20 minutes before time. So we have plenty of time to have some questions, perhaps. Do
we have any
something else?
Questions from the audience? Nan says, "Wait, it isn't magic." Nan says, "Well done."
Yay.
I'll actually be teaching about photosynthesis.
I do have something different, and it's great that we ended early, because I need to write a very strong worded letter to the Dutch Weather Channel, which I'm subscribed to on YouTube. Earlier today, they put out a video that next week we will have a full lunar eclipse. Not just a lunar eclipse, but a lunar eclipse with a blood moon. And then they told us that would start about 8 p.m., by which time half of the eclipse would already have happened. So I'm thinking it's not a full eclipse if we only get to see half. But the last half of the full eclipse, it's still a half eclipse. I'm going to write a very strong worded letter.
I think that's a good idea. Yeah.
I mean, first they tell you a full lunar eclipse with the blood moon. That's something I need to see. And then they tell you you only get to see half because half of the time it's going to be below the horizon.
Well, Peter, there's been a vampire.
That's
that
just
took
mean.
away half the blood.
Yeah,
so that's why you can't see all of it.
It's just mean. Telling us we're going to have that and then not.
So one of the lectures I'll get to give later in the semester, actually to both of my classes, believe it or not, is both of them talk about photosynthesis, but to kind of varying degrees. One of them goes really deep on photosynthesis in the biology class for science majors, and then the one that's the bio one for non-science majors doesn't go really as deep. That's a lot more surface level. But it talks about photosynthesis in the context of other phenomena. So in the context of the carbon cycle, and it talks about C3, C4, CAM plants and all that. And it also talks about, the chapter has a bit on how global warming will impact photosynthesis. And I, of course, use that as an intro to spend much of the class period talking about how we know global warming is true, and then also how people lie about global warming. Hello, Dragons Part 90 official. Welcome. Blood moons are the best time to channel feminine magic, says Smitty. I'll have to take your word for it. But yeah, so I get to talk about all the conspiracies around global warming. So
I find it very it's very strange, because it's the same type of people who will for example, when you discuss I think a good example is the origins of SARS-CoV-2.
Mm-hmm.
They will criticize you for just believing the experts, or just or trusting the government, even trusting the Chinese government even. But
Right.
then regarding climate change, some of the same people will then just say, yeah, the fossil fuel companies are right. I
don't think most of them know that it's the fossil fuel companies, though. I don't think they know that.
Yeah, exactly. You're right. Yeah,
that's
right.
one of the big.
It's ironic. It's ironic. I'm just saying. Yeah.
Ironic. Yeah. Because that's one of the points in Merchants of Doubt, was that they funnel the money through different organizations like Fox News or PragerU or whatever, the George C. Marshall Institute, to lie about global warming. And so it's you see, oh hey, a physicist is talking about how global warming is not real, so okay, maybe I should listen to him. He's an expert in this sort of stuff. There's actually, when we watched the one guy from PragerU, he was an atmospheric physicist, which is even more pathetic.
Yeah. It's also I've seen a recent publication by Professor Dave about
Matt Ridley. Yeah.
Yes, Matt Ridley. And he is also the one, he's also a climate change denier with a financial interest in fossil fuels as well. But we also recently published the book about arguing the lab leak, basically. And it's also a book promoted by John Perry as well. Yeah. Mm-hmm.
Unfortunate, yeah.
Yeah.
I really don't know.
It's funny you should mention this, because earlier
he wrote The Red Queen. I love that book.
I think a little earlier today, was it, that Dan posted the video that of
yes.
from this channel?
Yeah.
And 2 weeks ago, Natalia Reagan interviewed a guy who was instrumental with developing the vaccine for COVID, as well as the distribution.
I need
and he also mentioned that by now it's just a conspiracy theory that it was it came from a lab. So in hindsight we should have invited him for that conversation as well. That would have been funny.
Who?
So this is a guy,
the one who
Dr. Ian Bolton, who was in the Netherlands at the time, and helped develop the vaccine.
I mean, at first I wasn't gonna. Sorry, John had initially only wanted to talk to me, and then I was like, "Well, I'm not a virus person. I don't know enough about this topic." So that's why I reached out to Dan, and I didn't want it to be a dog pile. Essentially, I wanted John to feel like he could get out what he wanted to say in the show.
It would have been fun to have a scientist in the field present in that conversation.
Oh, sure. I mean, yeah. I mean, I'm happy to have Doc on again. I didn't want to overwhelm John. And so I was fine with the crowd that we had.
I had a conversation with him on the Peaceful Science forum a little bit. At one point, it went a bit downhill, but then I think we managed to get on good terms, although not on the forum itself, because he was locked out. Like he was a new member, and new members have a limit on how many posts they can post. So he was like, "Oh, can you help me with this?" And I will say, "Oh, I will help you with this, with the admins, and giving you full access to the forum and such." And then go, "Thank you very much." And all right, and I hope we can end on good terms. Sure. And that's basically how it ended, the conversation on the Peaceful Science forum. So I think it's very frustrating because I like John. I really respect his style of science communication. I'm just very disappointed. Not in the condescending way. Hopefully, I don't want to come over as condescending to John, but I'm just I did not expect this from him. Yeah.
I was also looking up
go ahead,
Matt Ridley. I was trying to remember, like I had heard his name before, and so I was trying to see what. And I have read The Red Queen: Sex and the Evolution of Human Nature, and I think it's a good book. Yeah. It's just about sexual selection and the impact of it on animals and stuff. Like that's all it is. It's a really good book, and so it's very sad to me.
Mm-hmm. And he also claimed that HIV originated from not the measles, the polio vaccine. Yeah.
Oh no. Really?
Yeah. Although he eventually did change his mind on that, but recently he made a tweet saying that, "Oh, maybe I'm rethinking about it. Maybe I was right before." Like, what the hell? All right, Matt Ridley is not a serious person.
Yeah, I mean it looks like, based on what I'm reading about him on his wiki page, he's a wacky far right libertarian also. So that is sad. Is what it is. But yeah, he wrote that book on COVID with Alina Chan, who's another one of the big.
Yeah, and she got on Jean Perry's channel with interviews.
Yeah. I remember
Alina Chan. I have not like a Matt Ridley is a bit very naughty, but I don't think Alina Chan has anything like that. She was just a very obscure researcher before pandemic. And she only got very famous on Twitter after the whole debacle.
Yeah. Sad. Yeah, it is pretty sad. God, I mean, it's really disheartening to see how many people became absolute cranks because of COVID. It is very disheartening.
it's not surprising. Like I can already imagine what it was like living through the Spanish flu. I can only imagine like that. Although the benefit of then is there was no internet or social media back then. That's one thing. But it's also one thing to mention, like the lab leak hypothesis is not an inherently conspiracy, like an
Sure,
inherently
it wasn't initially
pseudoscience. Yeah,
right. It was worth investigating.
Yeah,
initially,
and they did. Like you have like I forgot the names. Like uh, like one scientist behind the proximal origins paper, like I forgot his name. Like he seriously considered, like he was almost convinced about maybe they did come from a lab. Like but eventually, when they discussed the evidence and looked at it more carefully, he changed his mind. And of course, the lab leakers don't still crucified him, figuratively speaking, on this. Yeah.
Yeah, it is pretty sad. That
so
it's sad that it's so easy to become a crank and get paid exorbitant amounts of money to just do insane things. Professor Dave has done the stuff on like the Weinstein brothers, how crazy they've become. Matt Ridley, the guy who I forget his name, who's like a cosmologist, who's a very well respected cosmologist, but is now like a
Olsen.
No, not her. It's a guy. It was a man. But she is also nuts. You're right. Hassenfelder. She's also crazy. It's like Loeb was his name.
Not sure.
Hold on. Was it? Yes, Avi Loeb. Yeah, theoretical physicist. He's like aliens are real now. Hassenfelder's becoming more and more just like a far right mouthpiece of anti-science. It's guys, if this whole science thing doesn't work out for us, we could very easily change careers and just start lying. We could become like mega church pastors, or we can start talking about how science is broken online and how the scientists are lying about everything. It'd be so easy.
You'd make a lot more money, Jackson.
Right now. We would make so much more money. Oh my goodness, yes. We would make so much more money. Oh my gosh, the things I could buy with that kind of money.
If only we didn't have this stupid thing called ethics.
Yeah, that's true. If I had no ethics whatsoever, no morals, I probably could. I feel like I could do it. It'd be easy.
What I want to mention is like I said that the lab leak hypothesis is not inherently pseudoscience. What makes it pseudoscience is how the proponents respond to new evidence. Like I mentioned, the author of the proximal origins paper, they respond to evidence by changing their minds. But I have seen a recent internet debate between like somebody called Peter Miller. He accepted a $100,000 bet on debating the origins of COVID. And Peter Miller was he argued for zoonosis, and the challenger putting out this public challenge of bet $100,000 on this. And they got two judges to judge the debate. But Peter Miller won. The one arguing for zoonosis won. That's not the one I want to mention. What I want to mention is like one person, like one of the main proponents for the lab leak, participated in the debate, and at one point he basically said that even if we found raccoon dogs infected with the COVID, the early strain of COVID, you would think that that's pretty case closed. It's just a case closed case for the zoonosis origin. But no, he will argue that the raccoon dog was infected by humans instead of the other way around.
Right.
And when you respond to evidence like that, there is no point in arguing anymore. Like you will interpret the evidence however you want for your own narrative, and that is what makes it pseudoscience, the lab leak hypothesis right now.
Right. Yeah, it is just a conspiracy. You're right. Yeah, I mean, I'm glad we had that conversation with John Perry, and I'm glad that people are going to see it on Dan's channel. I am also glad I've noticed that the channel, the political streamer Destiny, is also now debating people because he was doing research into the origin of COVID, and he's also like, yeah, it was absolutely a natural origin, not a lab leak, and so that's also very good. It's good to see. It's good to see when really big streamers, come to the side of reason on this topic and whatnot. So that's good. But yeah, it was kind of sad. I mean, because we did our own video on this topic way back. How many years ago was that? That was like 4 years ago, 5 years ago, something like that. You remember Nestling?
Oh, sorry. Which video?
We did a video on COVID, like way back.
Yeah, that's like 5 years ago by now. Yeah, or almost 5 years ago. Yeah. I think that is pretty outdated because, like in that video, we mentioned the hypothesis about the pangolin intermediate, but that's no longer accepted anymore. Yeah.
Mm-hmm. Yeah.
It was like early on, like one of the closest relatives of SARS-CoV-2 was found in pangolins, but now we have found more intermediate. So now it's no longer accepted that the pangolin wasn't
right.
An intermediate, and also that was before we learned that the wet market housed raccoon dogs. Like before was at the point basically there's also a funny thing is that the Chinese government lied to us about the presence of wildlife in the wet market. At first they told us like, oh, no illegal wildlife, like nothing to see here. But then later we found out no, that they were there.
I was just looking at the video over on Dan's channel, and it's all the comments on it. I mean, it is. I think it's the first day, but I'm glad to see that all the comments are saying like, obviously it's natural origin. Sad to see that John is still pushing for the lab leak hypothesis all this time later. But yeah. I guess on this channel, I know we haven't published the next Ancestors Tale video, and we're like only 5 from the end or something like that. So we're very, very close. But part of the reason is I've been very busy out in the meat space. The Cauliflower's Tale is up next. It's actually, we finished it. We finished writing it. We finished editing it, all that stuff. I just have to make it into a video, and I just have to find time to do that because I've been writing my lectures like constantly for the 2 classes I have. So Smitty says, just go on Tucker Carlson. You're right. Or Joe Rogan. I just got to go on one of these channels and, we're set. It's done. It's over. Yeah. I mean, it's only like Cauliflower's Tail, then with the Redwood's Tail, I think the Hump Bladderwort's Tail, then like, the Mixotrick, Rhizobium, and then Tax Tail. Okay, so there's... So after we finish Cauliflower, there's 5 left.
Yeah. Also, the Smitty had one question about does HIV resemble poliovirus? And I was looking up. I'm not sure if poliovirus is similar to the HIV virus, but the idea like that which Matt Ridley proposed about the origins of HIV, he argued that the polio vaccine was contaminated with chimpanzee tissue, and that's how HIV ended up in the polio vaccine. But there's no evidence to back that up. Yeah. No, in any case. Yeah. But I'm not sure if HIV is similar to the polio virus. I'm not sure if they are in the same.
HIV is a retrovirus, so.
HIV is a retrovirus, right? Yeah.
Yeah, it's a retrovirus. So let's see, poliovirus is a. Let's see, it's riboviridae. So no, they're not anywhere near similar to each. It looks like, based on what I'm looking at, my very brief look at Wikipedia, they're not anywhere near similar to each other. It looks like they're both with the smallest classification that they share is the realm, which is greater than a kingdom, which is Riboviria. So they're very,
Yeah.
very, very different from each other. It looks like.
Yeah, and I don't think the polio virus does not undergo reverse transcription like the HIV does. Is a retrovirus, but polio is not a retrovirus. Yeah. At least as far as I can tell at this moment.
Yeah.
Yeah.
Alrighty. Well, thank you everybody for watching. Thank you, Nestle, for your presentation today. We enjoyed it. And we look forward to the next chapter of the show. And whenever I have the time, I'll get around to making the next Ancestors Tale episode. So thank you guys for watching. Thank you, Peter, for hosting. We will see all of you next time. So bye, everybody.
Bye-bye.